Okay, we're going to get started here today. Thank you all for joining us today for our webinar. My name is Jackie Carville, and I'll be coordinating the webinar today. I'm here with Matt Kaiser, who will be going over some of the functionality of DNASTAR's Lasergene Genomics Suite Advanced SNP Analysis capabilities. You may have noticed that your phone has been muted. However, we do encourage you to ask questions along the way. To ask a question, just type it into the chat dialog and select Send to Host. I will then direct these questions to Matt during breaks to be answered for the whole group. If you need any assistance or have any questions during this webinar, feel free to send me a chat message or email me at webinars@dnastar.com or tweet us at the Twitter handle @DNAStarInc. And with that, I'm going to turn it over to Matt here.
Thank you, Jackie. And I will, in a minute here, be sharing my desktop so we can start out with a little bit of a PowerPoint. Okay, so today we're going to be talking about some advanced SNP analysis in the Genomics suite. We've covered some similar topics in the past. Today I'd like to focus on some of the things that I didn't get a chance to spend too much time explaining or going into some more detail. So hopefully we'll be able to cover a few things and generate some questions from those of you that are attending. And as Jackie said, you can chat those in. I'll also be around at the end of the webinar so that I can answer any questions, and I can send emails at the end if we don't get to those questions today. So first, a little introduction to DNASTAR. We are located in Madison, Wisconsin, and that includes our sales staff, our development team, our support staff. DNASTAR, being centrally located, gives us some advantages. That means that we can be responsive to customers, answer their questions, improve the software in response to requests from customers. So it's a nice, again, nice central location to really support the software the way that it needs to be supported. Our history goes back quite some time. We've been a software company since 1984. Our founder is Dr. Fred Blattner. Dr. Blattner's lab in the mid-1990s sequenced the E. coli K12 genome. Some of the folks that participated and contributed to this research are still with DNASTAR, including Dr. Blattner and also Dr. Plunkett. And so we have long-standing expertise in producing software that is powerful yet easy to use. And of course, we've expanded our functionality beyond microbial genetics to all sorts of different workflows, and one of those that we'll be looking at today. So we produce solutions for next-gen sequencing data, Sanger data, all the different platforms, and multiple workflows. And our software runs on Windows and Macintosh computers. Our assembler will run on Linux, and we also have cloud options for those of you that would prefer to run and do your analysis up in the cloud rather than on local hardware. So our software is research grade, so it's meant for researchers to utilize and to incorporate in their publications. And if we do a comparison across the literature, and you can see that DNASTAR, dating back all the way to 1985, has been the most cited sequence analysis software. And so the software is research grade, yet it's going to be easy enough to use, even for some of the more complicated next-gen sequencing projects. And I'm going to show you a project setup, how easy it is to do that, as well as the downstream analysis. So one of the first questions that we'll get from customers working with next-gen is really questions regarding the performance. There's many different assembly options out there, and many of them require high-powered Linux boxes or clusters to get acceptable performance. And our software is designed to run on much more modest hardware and without sacrificing performance. So I'd like to show some of the assembly times that we get with different data sets. And again, this is just a subset. We have more benchmarks on our website that go into more different project types. But to give you an idea, human genome assemblies, this can be over 1 billion Illumina reads at 36x coverage. Takes about 13 hours to assemble. The data sets that we're going to look at today are exome data sets. And depending on how many you pool together, the project can be 1 exome or multiple exomes. It takes roughly 2 hours per exome, actually a little bit under that when we batch projects together. And so that's a very, very good assembly time. And that's also, I should make a distinction, is that we call this assembly time, and it really is the assembly, the gapping, the statistical SNP calling and filtering is done, as well as writing out all the output files. And so this assembly time is not just the mapping time, but it's the entire, from when you press start to when you have a completed BAM file at the end. And then other data sets that are smaller, like targeted resequencing panels or RNA-seqs, they may take just a few minutes to an hour, somewhere in that range. So again, this assembly time is extremely competitive. And again, what the key distinction is the hardware that we're running the software on. So you would expect assembly times to be very fast on Linux clusters that cost tens of thousands of dollars. We design our software to take advantage of the hardware that is accessible to most researchers. And so in this simple diagram, we can see that a computer that has between 4 and 8 cores, it doesn't really benefit to have more than 8 cores, and 16 gigs of RAM is a sufficient core computer. And then you need some storage space. The data files that you're assembling for exomes can very easily be, a few hundred gigabytes of just raw FASTQ file data. And then, of course, the output files, the BAM files, those can be multiple gigabytes as well. So a nice storage disk, 1 terabyte, 2 terabytes of storage is usually sufficient. And then there's one other piece of hardware that really is important for the performance of the assembler, and that's a dedicated hard drive for handling the temporary files that are generated during assembly. And so we call this a scratch disk, and it varies in size depending on how large your data sets are. And so if you're doing single exomes or just a handful of exomes, a 1 terabyte disk will be sufficient. If you're doing large batches of exomes or human genome assemblies, as much as 4 terabytes of space would be needed. And this disk can be internal, so if you have a desktop machine that has room for additional drives, it can be an internal. Many customers opt for plugging in an external drive through USB 3 or eSATA. If you have an SSD drive, you get better performance. It reads data faster. So again, a computer like this might cost a couple $1,000s, even under that, so you can get this very good performance on budget hardware. So our software, in addition to being powerful, is also supported very well. If you go to our website, there is a support section, and under training and support, we have webinars like we're doing today that we post up on our website that are on a variety of different topics. And then we also have shorter training videos that can be several minutes in length. And the training videos can be next-gen workflows or just basic, priming, cloning, sequence editing. And again, if you have questions, you can refer to some of these videos, and then also just contact DNASTAR, and through email or phone. I often set up personal webinars, so if you're working on a next-gen project and you have a lot of questions, we can schedule an hour or two to go through your project in detail. So again, great support, powerful software is some of the key attributes. So we're going to be talking about the Lasergene Genomics Suite today. The Genomics Suite is what our next-gen customers will be using. There's 3 components that we'll be talking about today. It's the Seekman Engine Assembler, Seekman Pro for visualizing the assemblies, and then ArrayStar for doing some of the downstream analysis. There's other suites that we offer, the Structural Biology Suite, the Evolution Suite, that do not include the next-gen assemblers, but may focus on some other areas like protein analysis, NovaFold, which is a structure prediction program that's brand new. So there's a number of other functionalities that we aren't going to cover today. And in fact, you can buy, in addition to a genomic suite, a complete DNASTAR suite that would include protein tools as well. So again, if you're interested in the software, we can provide you with all the details and the different options that you have. But today we'll be focusing on the Lasergene Genomics Suite. So the Genomics Suite supports a wide variety of different assembly and analysis workflows. And we're going to be focusing today on exome and targeted resequencing. And we'll be looking at specifically some exome data sets. But if you're doing multiple different types of projects, it's good to have a software that can handle a wide variety of different projects. And that includes RNA-seq. We do de novo transcriptome, de novo genome resequencing. We have some specialized workflows for microbial genomes where we can auto-resolve some of the newly sequenced genomes. And so we have this flexibility built in to allow for different workflows. Today, though, we're going to be looking at a data set that was downloaded from the short read archive. And it's a data set that consists of 23 prostate cancer exomes. So this is just publicly available data, and there's a little bit of information here. There's a set of genes. TP53 is one that is present in 5 out of 16 samples. So it's a nice data set that we can load into the software, find lots of variations, and maybe compare it to a control set. So the goal really becomes sorting through all the background variations. And if you do whole human genome assemblies, that can be as many as 4 million background variations per individual. When we do exomes, of course, that's going to be a much smaller scale, but it can still, very easily be, 100,000 variations. And what you're trying to find is the variation or the small set of variations that are related to the disease of interest. And so it may be a SNP analysis, it may be some genes that were a gene set that is common to the disease group. And so what we hope to do with the software today is to show how do we get from sequence data that contains thousands or millions of sequencing artifacts mixed in with real variations, and then try to get those interesting variants to drop out after we filter through the data. So that's kind of the goal today. And so the kind of workflow that we're going to go through in the software is starting with cancer and control data sets, and we can load those into Seekman Engine. And Seekman Engine we'll look at some of the auto SNP filters that are built into Seekman Engine and how we can control those. We take the output project and look at the manual SNP filters, so we can apply additional levels of filters that are interactive. We can create a user SNP database in Seekman Pro, and again, we can look at, verify in Seekman Pro, and then once we've done the verification, we can import the SNP information to ArrayStar. And ArrayStar allows us to pull in additional information from SNP and gene annotation databases, and then we can create SNP sets and gene sets and do association studies between different groups. And with ArrayStar, then, we can identify the candidate genes and SNPs for our disease group. So with that, we'll jump out of the PowerPoint and go into the software. So I'm going to launch Seekman Engine. And so Seekman Engine is a 64-bit program. It runs on Mac, PC, and Linux. What we're looking at here is really a wizard, and the wizard is what allows us to help set up the assembly. And it's designed to have a flexible interface so that I can create a new project. I can select the project type. So if I have a genome or a targeted resequencing, when I select targeted resequencing, I have one option for an output. It's going to be a BAM file. It's not editable, but it's unlimited in the amount of data that we can load into it. Just by comparison, if we selected genome assembly, we have lots of different workflows. De novo, we have a hybrid de novo templated workflow, and we have a legacy workflow that we offer. So as we make choices in this wizard, we have different options in the following screens. And with these options, we'll have optimal parameters that are put into play. And so the idea here is we make the wizard very easy to use, and we can select an output folder and also temporary file location. And the temporary file location, again, is our scratch disk. It's very important for processing our data. So we want a good, fast drive with lots of space. We can add our template files. When we're doing targeted resequencing with a main model organism like human or mouse, it's a good idea to download a genome template package. And a genome template package is available from the DNASTAR website, and the package includes dbSNP, COSMIC, and GERP database information. So we take this database information and we bundle it together with the GenBank annotated chromosome sequences, and that can be downloaded together. And you can see here we have a list of organisms. And if we go to human, we see there's actually a number of different human genomes that we can analyze. There are 3 ethnic groups, a European, Asian, and African ethnic group. We have different builds. So this first column is the dbSNP build. So, and these are updated on a fairly regular basis. So right now we have 137 available. We know that there's build 19. We know that we have the next human genome build coming out very soon. And so, of course, this will be under a constant update. Because some folks have done a lot of analysis in older builds, they may want to maintain that older coordinate space. And so we make the older packages available so that you can do that. And so you can see we have a number of different model organisms here. I highly recommend downloading this package and using this as the reference. The advantage to that, of course, is that you get all this database information then incorporated into the analysis. And so it really facilitates your analysis. So I'm going to go and grab my genome template package. And what Seekman Engine can also do is that if you have known dbSNP RS entries, so as Seekman Engine processes this genome template package, it can start to do statistical SNP calling then and use that information where known SNPs are, and that can influence the probability calls. So if it's a known position, you can have a stronger probability that the variant is real at those locations versus an unannotated genome. So there are some definite advantages to feeding this information so it's present while the data is streaming through the assembler. So it makes for a much more efficient analysis. So all the SNP calculations are going to be pre-flighted and done at the assembly time. We can then add in sequence files. From the pull-down, we have our different platforms, so we can select Illumina data or 454 or Ion Torrent. And we can load both paired and unpaired reads. I'm going to just grab some data here just to, not quite where I thought it was, so we'll just go up here. And I'm actually jumping up to my network here, and this is something that it's okay to do. If you have a nice fast network connection, you can go and grab data here. And I'll just do this again. And these are different samples. These are SRA files as well, so different exomes. I'll just add one more. And of course, the full data set, we'd load all these FASTQ files. And you can see that for one sample, we've got, this is a very deep coverage of this exome. You know, these are multiple 2 to 5, 2 to 7 gigabyte files. And I have a program installed called Universal Viewer that allows me to peek inside these really large files. Don't try to open these in Notepad. It won't. They won't open in Notepad. But I can take a look inside the file. And if you're just getting started with this sort of data, it's good to have this Universal Viewer. It's a free download. I can see that I have a Illumina formatted file. I have a name followed by a sequence, followed by the name again, followed by the quality scores here. And so it's a great program just to look inside. Sometimes it's confusing as to what file you should actually be using. And so this. Okay, so I've loaded in some data. Now I have the option, and this is important for your analysis, but I have the option to take multiple exomes and assemble them together and create one BAM file that has multiple exomes in that file. So, for instance, if I'm looking at 3 or 4 exomes, that might be nice to be able to see them aligned together and do the comparisons. However, if I'm doing 100 exomes, at a point, 100 exomes is too much in one BAM file to really efficiently do the analysis. So we've got a couple of options. I can tell the assembler this is a multiple sample data set, and what Engine will do is look at the file name and assign that file name as the group name, which I could edit. I can customize this to whatever I want. And I can also assemble these as separate projects. So in this case, if I run as separate projects, I could load in my 100 exomes worth of data and run and group them by name. I may have multiple files per group. And this will produce 3 different BAM assembly output files. And so it's a really nice way to automate what in the past we'd have to use a script or to kind of manipulate how we ran the assembly. And so this is a nice new tool. There are some assembly options now. The minimum match percentage really impacts the assembly more than anything else. If I make this number higher, it makes a more stringent assembly. And what that means, at 97, a read must match the template or the reference genome 97% in order to align. So if there is one thing that you would change, it would be minimum match. And usually it's to make it more stringent. It's pretty rare when we actually want to make it less stringent, but we'll go from the default, maybe go a little bit higher, 95, maybe 97. So now for SNP calling, there's a number of things here that we need to consider. The genome ploidy, we give a distinction between haploid and diploid or population. Doing the p-values for SNPs depends on whether or not it's a haploid or a diploid, and then you can get the correct genotyping call and get the right confidence scores. If you have a mixed population, then you pick population or other, and we don't apply the same statistics. It becomes a set of simple filters and manual filters. If I leave this on diploid, we'll go to the advanced options now. And in the advanced options, we have some layout options, more knobs and dials with alignment, but we want to focus today more on the SNPs. So in Seekman Engine, there are, by default, we'll calculate SNPs, and we can set the method to be diploid, haploid, Bayesian, or simple percentage. And you'll notice that by default with diploid, there's some filtering that's going on here. One of them is minimum SNP percentage. So what these filters are used for is they are upfront filters that will remove much of the sequencing noise. So all of these next-gen data sets will have a significant amount of artifact due to errors in the sequencing. So we apply some minimal filters that can remove the sequencing noise from the SNP files. At these scales, when you have millions of SNPs potentially, the SNP files can be pretty large and unwieldy. And so to make the projects easier to handle and the SNP files a little bit smaller, so we automatically filter SNPs that occur in less than 5% of the aligned reads. If the p-value is below 10, so really low values, there has to be at least 2 variant bases, and the minimum quality score has to be 5. So it's very, very minimal filtering. And we can also check strandedness. That turns on a part of the algorithm that is aware, is the SNP occurring always in the same strand, or does it occur on both sequencing directions? Some sequencing artifacts, depending on your technology, you'll see only in one direction. When you sequence one way, the SNP shows up but not the other. That's usually an artifact of some sort. If you have deep assemblies that are over 25 to 50x coverage, turning on the strand awareness can be a good idea, and especially if you have a lot of noise in these sorts of areas. It's turned off by default because when you have thin areas, it can become a little more of a gray area if you've got areas that are 5x or 10x coverage. And so these are adjustable, so I can reset these values. So again, these are manual overrides. So if you have a data set where you're interested in SNPs that occur at less than 5%, I can change this to 0 and allow those SNPs to go through. Now I may also want to affect my confidence threshold as well. So I can completely remove all the filtering as well. So it's good to know that this filtering is occurring. If I'm in a data set of mixed cells, or where not one genotype or a polyploid genotype, we don't have the p-values calculated, and we just have a SNP percentage, and I can set this to 0, and this will allow every sequencing artifact and variation to show up in the SNP report. Okay, and then I'll have to manually filter downstream at the next set of filters to remove those and to separate the signal from the noise. So this is a very good thing to know about. There's also there's some help here that's contextual that will explain in more detail what some of these things are doing. Okay, so I'll go back to diploid here. When we click Next, the assembly is ready to begin. And what you see here is a script that's been written. And the script is the instructions to the assembler of all your parameters, the file pathways. And when you click to assemble, this will automatically save a script file out. So it's a little text file that is these instructions. And when you click assemble, if everything's working fine, if you're here's the scratch disk, and I've got about 6 terabytes on this computer. If that disk isn't ready, we might get an error right away. And you can watch the progress of the assembler. And if there's a problem, you can export the log, send that to DNASTAR, and we help troubleshoot that for you. We also look at the task manager. The task manager will show us the CPU usage and the RAM. Seekman engine is called XNG when it's doing a templated assembly. And you can see that for a burst there, it was using 97% of my CPU. This is an 8-core computer, and so in short bursts, it will use almost all the processing power. And so the software is optimized to use as much hardware as you're making available to it. Now, that said, I haven't seen XNG use more than 8 cores, even when more than 8 cores is provided to it. So I don't think it makes any difference to go above 8 cores. And 16 gigs of RAM is more than enough for exome assemblies. But it'll keep this computer busy at least for the first half hour. Then it settles down, and I can do other things on the computer. So I'm not going to let this run during our webinar, because we might start losing our sound quality. When it's done, we'll have a button that says Open in Seekman Pro. So I'm going to quit my assembly here. Just double check, make sure it's stopped there. Okay, we're good. And Seekman Pro is part of the Lasergene core suite. And Seekman Pro allows us to do all the visualization and to look at the assembled data. And so the first window that's open will have a list of the contigs. And in this list, you can see NC_0001. This is human chromosome 1 and chromosome 2 and 3 and so forth. Some of the do we have a question?
Yeah, Matt, before we get a little further into Seekman Pro here, we do have one question on Seekman Engine. So the genome template packages that we looked at earlier in importing, what if somebody's working with an organism that isn't in the DNASTAR list of genome template packages?
So if you're working on an organism that's not present there, you can request that a package is constructed for you. And what we'll do is we'll go to dbSNP and first verify that there is the SNP information is curated there, and there's enough of it there that it warrants making a package. If you do not have, if we say, well, there really aren't the SNPs curated at dbSNP, you can create a package, and I can show you that here with Seekman. We can create a genome template package from scratch as well, and so that would be a good solution for you.
Perfect. Thanks, Matt.
You're welcome. So Seekman Pro is a 32-bit program. You don't have to run this on a super powerful computer. It's designed so that I can run assemblies on my more powerful computer, do my analysis on my laptop or whatever computer I have the easiest access to. So a project like this that has 3 exomes open, 3 exomes in one project, you can see it's using under a half gig of RAM and very little CPU to keep that project open. And so what's nice about BAM files is they allow us to open really big assemblies but keep a really small memory footprint. And so I can do this analysis then very efficiently, and Seekman gives me a really nice interface for navigating through the assembly and looking at these different regions. So the genome template package will have all the chromosome sequences. For some packages, there's also some unlocated or unplaced variant sequences that do not fit in any of the chromosomes. So you might see a longer list of some smaller, shorter sequences, that's due to those unplaced or unlocalized sequences. And so this is just a list of the contigs. Typically, when we go to navigate, we'll go to the SNP report as kind of the first place. The SNP report contains lots of information, and the way that it's structured is the upper header here contains the set of filters that we can apply. And I know it's pretty small here, but you can see the number of SNPs that are in the project right now, and each column then has a different bit of information. And so, depending on how you filtered up front at the assembly, if you removed all the filters, for instance, and just let everything come through, you could very easily have 10 million sequencing errors. And so this number will be really big, the SNP report will be gigantic, and then you have to manually apply some filters to try to reduce that size. And so I watch this number and then apply filters. I have some filters applied here already. Right now I'm just showing the subset of SNPs that create a coding change. And right now I'm showing all SNPs, but I could easily apply a filter. I might decide that I really only want SNPs where my depth of coverage is at least 10. I might also say the P0 ref, the probability, I want that to be 90. And the number just changed a little bit so far. And so actually this is 10% to 100%. My depth is over here. I clicked on the wrong one there. So here's all the SNPs have a depth of 10, p-value of 90%. I can type in a coding feature distance, so I could say, well, show me all the SNPs that are in a coding region or a certain distance from a coding region, maybe 20 base pairs. So I can apply these filters, and this number is going to change. I can also say, show me only the novel SNPs. So the novel SNPs are SNPs that do not have a dbSNP RS number. Or I could say, show me only the annotated SNPs. And now we're going to have either a dbSNP RS number or a user ID. And I'll explain what that is in a moment. So these are SNPs that have some sort of annotation in a database. So we can apply filters and get a result here on, a live update of that result. Now the SNP report is interactive with the alignment views as well. And so if I double click on a SNP, it can bring up that alignment view. And again, this is a 3 exome project, and I'm just collapsing everything down. So the alignment view then shows me the reference sequence. And if I click on the triangle, I can see the annotations that are at that location. Here's the 3 samples, the 3 exomes. And I can see from this view that in the reference sequence, these positions are colored green, and that means that they are annotated in dbSNP. And I found SNPs in all my samples. If I want to look at an individual sample, I can expand the sample and then look at the aligned data, and the SNPs stand out as blue highlighted residues. So again, it's a great way to navigate through the alignment via the SNP report. Now the columns contain lots of information, and so I'll kind of scroll here to the left. We have the first column, which is the verification. Has this SNP been confirmed? Right now we have most of the SNPs in here are unconfirmed. So if I look at a SNP and say, well, that's a real SNP, I want to make note of that, I can click select, or I can select multiples and confirm. And that means I've looked at these SNPs and I've decided that they are in fact real. When I do that, I can start to build a custom SNP database as well. And so if I confirm SNPs and then go to SNP append checked user SNPs, what this is going to do is add these SNPs to my little database that I've been building on this project. And so this is a little peek at that very simple database. And it tells me which contig, the reference position, It gives me a dbSNP number if there is one, and then it assigns a user ID number. So oftentimes these are done to add SNPs that aren't in dbSNP. They're novel. We've verified they're real. We want to make a record of them. And it tells me which assembly that these were built in. Now, if you, we had the question about the genome template package. If you didn't use a genome template package to start, you'll get a window that says that you didn't have a genome template package. Would you like to create one? And then you can say yes, and it will auto-create a package, and then it can write this custom database file. So this file is being written down. So here's the genome template package that I used for this assembly. So in the dbSNP folder within it, there's this data.other file. And this is my custom database file. And if I look inside, I can see all my information here. So if you have an organism where you don't have a genome template, it'll create the template and then write this file down. And so this file then is keeping record of my custom SNPs that I'm identifying. Now if I delete this, then I lose my user SNPs. If I move it, so it's something just to be to recognize that if I want to use this over and over for my projects, I want to maintain this genome template plus the data.other file that's within it. And now it's asking me to save, and so now I just save this selection of SNPs to my file. So the other columns that we have here, we have the MID, which is the group name from my multiple sample assembly, the contig ID, which tells me the chromosome number, a gap coordinate, a reference coordinate. Now, for this kind of a project, I'm not as interested in the contig position, the gap coordinate. So I can customize these columns as well. If I right-click and do a show hide column, I can go through and pick things that, maybe for this data set aren't quite as interesting or as relevant. So contig position are not quite as relevant. I'm not as interested in splice, homopolymer. QCall, which is actually a little confusing. It's really a quality call or a confidence in the genotype call. So I can turn off a few of the views or a few of the columns and kind of condense, because this table gets quite large, so I can condense it down a bit. We distinguish whether the SNP is a indel or a substitution. And if I hover over this point, if you're wondering why is that colored pink, and if I hover over a point, it's because it's magenta because it causes a coding change. And so that's just a little bit of a color cue there. We get the reference base, called base, genotype, the impact on the protein, the percent that the SNP occurs. I'm just going to scroll to the right here. The probability that the aligned bases are not the reference, so that's a p-not ref. The dbSNP ID, so we get an RS number. I can right-click here and open dbSNP. And so it's going to use that RS number automatically and go to dbSNP and look at the SNPs and all the information about that particular variant. I can also do the same if I have a cancer database. So here's a cosmic, and I can, let's see, I'll show that in the RASTAR actually, but we have a cosmic score, and then GERP score, which is an evolutionary rate profiling score for how conserved those residues are. We get user IDs. So these are all the SNPs that are already been identified and are in our custom SNP database. Feature type, feature name. We have a DNA change, amino acid change. So really lots of information here that we can use. And so Seekman can be used then to go and really confirm SNPs. And you need to look at the assemblies and double-click, bring you to that point in the assembly, and do the visual confirmation on that assembly. There's some other places that we can look, and I'm just going to go to a strategy view here. So this is a genome browser type view. There's also some variations, and we'll go back to the type here. So these are substitution SNPs, but we also have, double click again here, we also have some indel variations. And indel variations will show up in the SNP report as long as the sequence read, the indel isn't large enough to prevent the reads from aligning. And so this top one actually sorts out pretty well. This is, we look at this top series of indels, we have an insertion of 3 bases here. So if we double click and look, I'm just going to collapse this down here. So the real small indels, again, depending on the read length that you have. So one of the samples has a CCT insertion at this point, and the other samples do not have that insertion. So we can pick this up very cleanly and go in and look at, it looks like it's heterozygous, and so we can go and look at that particular insertion. If it was a little bit bigger with this sequence size, we'd get reads that don't align in that area, right? And then it's not going to show up in the SNP report. So we do have another report, and it's just important to know that that's kind of a gray area in the sense of it matters what your assembly parameters are, your read length and read technology, whether or not the indel is here in the SNP report or in this other report that we have, and that's the structural variation report. So if the variation is large enough to inhibit assembly, we'll get a list of insertions and deletions and indels in this report. And so, let's see, I'll have one that's, so to give you an example, here is a deletion that is 12 base pairs long. And so I can look at this deletion, in this alignment view. The strategy view really is a great place to look at deletions. And the strategy view, I have it set with this little key to show me only the reads that are split that flank the deletion. And so we get these purple or pink reads, and I can see I get a drop off in coverage, I get split reads. This is clearly a deletion at this point. So it's a nice thing, for the end users to see that we can pick up very small indels in the very smallest in the SNP report. When they get a little bit bigger, we pick them up in the structural variation report. So Seekman is great for looking at a handful of samples. When I want to start looking at larger groups, ArrayStar is the place to go. And ArrayStar allows me to associate and say, for example, if I have a cancer group and a control group, can I compare the SNPs between the different groups? Can I find a set of genes that contain variants of interest between the 2 different groups? So ArrayStar allows me to import experiments. So if it's SNP analysis and I click on import SNP, I can add my Seekman engine.assembly folder. So the same file that we looked at in Seekman Pro, we can bring into ArrayStar. I could bring in a folder that contains 100 exomes. So ArrayStar has a very large capacity, so I can bring in very, very large data sets. And the first place that we usually look is the SNP table. And so this is an unfiltered SNP table. So we've got lots and lots of information here. And we can see we have lots of columns of some different information that we maybe didn't have before. And the way that I got all this information is by importing a VCF file. So information like minor allele frequency, function, cDNA position, Polyphen, clinical associations, oftentimes that information is contained as annotations in a VCF file. And so I can import my VCF files as annotations now. And so here's a VCF file from the exome variant database, and I can take a peek inside of it. And these vary a little bit, so depending on where you get them, there's going to be some differences in the header. They haven't been fully standardized. And then we have all the information about the variations. And so ArrayStar will allow me to select my VCF file. It'll bring up a custom wizard then that will allow me to define what the different columns are if needed, and then extract that information. Once we've brought this information in, then we can filter now. So I've got all these SNPs, and I can use this information to help me find SNP subsets that are interesting. So we have a filter button here. And I can filter on gene levels or SNP levels. And so I can combine filters. So ArrayStar gives me all these filtering options. And I'm just going to show you a simple example here where I can do a SNP search, and I can search for SNPs in my cancer group and then set some criteria. Maybe non-synonymous SNPs, and again, this is where you want to spend most of the time in ArrayStar, is just trying out different filters. And maybe I want just homozygous, those that are definitely going to have an impact. And so I can search how many SNPs will meet that criteria. So this should be a pretty big number. So we have 2,445 SNPs in the cancer group that meet that criteria. But now I can add additional, and I can say, well, let's look at these annotations. And I could look at Polyphen, for instance, and say, show me all the SNPs that have damaging in Polyphen that also meet this criteria. And now we have 248 SNPs. So I might decide, well, those I'm interested in. They're non-synonymous. They have been linked with possibly damaging in Polyphen. I could go back and say, well, I can make my criteria, no start, no stop, nonsense, or frame shift, and redo the search. And that's what's really nice about ArrayStar, is the power and the ease of use of all the filters that we can apply. Or I might decide that rather than Polyphen, I might want to go into clinical association and just find all of them that are in the OMIM database. So I could say find any SNP annotation that has OMIM present. So again, I'm using those annotations that I ported in from the VCF file now to help me search and find different groups of SNPs that are potentially interesting. And so now we have 49 variants that meet that criteria. And you'll notice the results are listed below, and the results contain links. So here's a list of my genes. There's a dbSNP link in ArrayStar, so I can go right to that SNP in dbSNP. All right. I can keep scrolling here. I get GURP scores for some, and Polyphen. So it's all the information from that VCF file will populate. Then I also get the clinical association, which is the OMIM link. And so I can go and see the database in OMIM. And the same thing if I had cosmic information here. There can be a cosmic column and a link directly to cosmic. Let's see here if I've got, and here's a cosmic link. So again, we're linking back to the databases so we can find the information regarding that SNP in these databases. So it's a great tool then for doing wide-scale associations, and then pulling in database information to help with the analysis. Now, once I've done searches like this, at this point, I can start to say, well, I want to compare different sets, control versus cancer. I may want to do a gene-level search. So in some cases, just like the SNP information, I may have gene annotations that I can port in. And I'm just going to close this. So I can also bring in download annotations at the gene level. And so here's a list of databases for human. We have the Go annotations, and these have already been downloaded into this project. And so I can pull them in. So then if I do a gene level analyses, I can get a gene list, and these are Go consortium terms. So ArrayStar allows us to search at the SNP level to find SNPs, but then also to say, here's my set of SNPs that are interesting. Now, which genes are involved? So I'll just go back here to a set list. So when I've done my searches in my SNP table, I can save those sets, And with those sets, I can have as many as I want. So I can have control and cancer genes, and there's some quick links, some actions here where I can show a table of those genes. I can also go to places like Venn Diagrams and create Venn diagrams from the sets. And so in this case, we have 220 cancer SNPs and some SNPs that they share in common. And I can focus on the different intersections here and create gene sets or SNP sets and do additional analyses. So a great set of tools. Again, the goal with the software is to go from large data sets with millions or billions of sequences, and you may have millions or thousands of SNPs, and then be allowed to quickly filter through and find just those sets of SNPs or genes of interest that may contribute to the disease conditions that you may be interested in. So it's a nice set of tools. If you haven't used the tools at all, I highly recommend contacting us for demos. They're fully functional. And I'll just do go back to the PowerPoint here real quick and just kind of sum things up, and then I'll be available for questions right after as well. So just kind of a summary of what we've talked about today. The Lasergene Genomics suite assembles next-gen data from all the different sequencing platforms and supports a wide variety of different project types. This is software that runs on a desktop or the cloud. The hardware required is minimal. And then the variant calling uses Bayesian statistics to get p-values. There's lots of different filtering options at different levels, up front during the assembly, and then downstream filtering options. And we can pull information in then from a variety of different sources and databases, including a custom database as you begin to annotate your own custom SNPs. And then there's additional filtering and statistics and pathways that we can analyze in ArrayStar for our SNPs and genes of interest. So again, thanks for joining me today. I'll be happy to answer any questions as they come up. Thank you.
Sure, Matt. Yeah, we do have a couple questions here at the end. So the first question we have is, suppose that someone selected, 100 SNPs for SNP genotyping, and you want to design primers. So how would you go about doing this?
Right. So if you're in Seekman and you find an area where you want to design primers, you can export sequences from Seekman to our Lasergene Core, our Seq Builder software, and Seq Builder then can design primers flanking any point of interest. So you'd be able to use our Seq Builder tool to do that.
Great. And then another question we have is, so you talked about using different VCF files. Where can someone go about obtaining a VCF file, and what if there's no information or specific VCF files for the species they're working with?
Yeah, so VCF files are I'll call it a loose file format, so they aren't strictly defined. They have header information. At the very minimum, they need to have a reference sequence, a reference position, and the variant call. So I think it's 3 columns of information. And so VCF files for human data sets are usually from the exome variant server at WashU, but there's also 1,000 Genomes project for humans. The other VCF files could be produced just about anywhere, any other kind of analysis software. So if you're working on a non-model organism and you have some VCF files, what you do is you bring them into ArrayStar, and ArrayStar will recognize that there are columns of information, and hopefully it'll just read the VCF file directly, but you can also use that custom import wizard to define what the different columns of information are to bring the data into the project.
Great, thanks, Matt. So we have a couple other questions floating out there, but it looks like we're almost out of time here. So I'll make sure to get to your questions following up with you via email after the webinar. In addition, if you think of any other questions later, you can feel free to email those to me at webinars@dnastar.com. Also, feel free to email me with ideas for future webinar topics. Thank you again for joining us today. We hope you found this webinar helpful. As Matt said, we do have a large collection of videos on our website, so I would encourage you to check those out when you have some time. In addition, you can watch the recording of any of our past webinars, as well as register for any of our upcoming webinars on our webinars page. We are currently in the process of scheduling additional webinars, but you can expect a webinar coming later this fall on various alignment workflows. We hope to see you there. Thanks a lot.